So let's do a summary of epigenetics and directory programming, and then we can move on to the next unit, which is going to be cell structure. So the big emphasis there was that the epigenetics of a cell dictate its state, dictate what kind of cell it is. And so if we don't erase all of the epigenetic information in a given cell, you might end up with kind of artifacts in the final cell that you create, which don't in fact manifest as a healthy cell. So the example we're going to use is neurons in aging patients of varying levels, and we're going to see what the effect that has. So if you have your neurons and it's got your dendrites and your axon, and that's all very exciting, and you take that neuron cell, or you take a fibroblast from an aged patient, and you directly transform that fibroblast into the neuron, and it so happens that the disease that neuron has is an aging-related disease, and it's controlled by epigenetics, and you don't erase all of the epigenetic information from that fibroblast, your stem cell therapy, or your cell therapy in this case, is actually not going to help at all. In fact, it could make it worse, because we don't know what effect that has. This hasn't been tested, we haven't actually done it, but for the case of argument, for the case of this discussion, we need to understand that without erasing all of that epigenetic information, you could end up with cells that behave in the exact same way. And so the solution to that is to, in situations when we think we could have epigenetic issues, is to de-differentiate fully into induced pluripotent stem cells and then re-differentiate into the cell type that we want. So, of course, we have our stem cell and then back up to neuron. Cool. What is going on with the focus? Alright, that should behave. Okay, so what are the big concepts of this section? Alright, so cellular identity is regulated by epigenetics. Cellular identity is regulated by epigenetics. Differentiated cells don't like to become induced pluripotent stem cells because there are mechanisms that lock them in place. Okay? Differentiated cells are locked from becoming iPSC. Okay? A way that we can induce that kind of activity is with exogenous transcription factors. Transcription factors can cause these cells to de-differentiate or change into other kinds of cells. And that's important for us because we want ways to force cells to turn into the kinds of cells that we want. Now, there are particular kinds of exogenous transcription factors called pioneering, which have the ability to bind to closed areas of chromatin. And we mentioned this a couple of videos ago. And that's very important because unless you're able to open up that closed chromatin, you can never reprogram that area of the genome. And you need to be able to reopen that area of the genome. And finally, induced pluripotent stem cells, iPSC, erase epigenetics. iPSC arrays epigenetics, and that's kind of gone downwards a little bit. Right, so that's cool. Now, what's the point? What's the point of all this? What's the point of having direct differentiation or non-direct differentiation? What do we achieve by that whole thing? what we achieve is we can use it for stem cell therapy, tissue therapy, so we can replace damaged or diseased tissues. We can study the differences between the iPSCs and the directly differentiated cells, and we can understand what they're useful for. And we can use these cells for drug examination or disease in a dish kind of approaches. So for example, if you have your directly differentiated cells and you want to test them for that disease, and that disease is actually epigenetically regulated, you can use those cells directly. Okay, so that is direct differentiation and genomic predictive reprogramming. We've also discussed today the big concepts of epigenetics and mogrify, and also the applications of these cells. Thanks, guys.